Let's talk about the DNA replication fork, a high-yield topic in genetics, biochemistry, and on the MCAT. We're going to cover the high-yield enzymes, and we're going to focus on concepts that I think a lot of students get backwards, specifically the leading versus lagging strands, which one starts with 5', which one starts with 3'. Rather than just telling you the facts and hoping that you memorize them, I'm going to explain the underlying mechanisms behind the concepts so that you don't have to rely too much on memorization. Let's get started. Before we get started, I want to remind everyone, if you haven't yet subscribed, be sure to subscribe to stay up to date with all of my MCAT content. Also check out my MCAT Biochemistry playlist and MCAT Lab Techniques playlist in the description box down below. The first enzyme I'd like to discuss is helicase. Helicase's job is straightforward. It's represented by this red triangle over here. Its job is to separate the two strands. The second enzyme we're going to discuss over here is DNA topoisomerase. It's represented by this green circle over here. The function of DNA topoisomerase, sometimes also called gyrase by the way, the function of it is to prevent the supercoiling of the double helix. So as helicase is doing its job and it's separating the two strands, you'll start to see that these will coil on one another over here. So the function of DNA topoisomerase is to prevent the coiling of the double strand over here. Also, just as a side point, not something you need to know for the MCAT, but topoisomerase is a target for certain antibiotics and for certain chemotherapy, i.e. cancer treatment drugs. Our next enzyme is represented by this purple circle, which probably doesn't look purple because the color is smeared. But in any case, it's represented by this circle. And as you can see, I've represented that it started to lay out some of those nucleotides. Now, this isn't DNA polymerase, which is responsible for replication of DNA. This is actually primase. And the reason why we start with primase is because DNA polymerase cannot lay out the first few nucleotides. It needs someone else to come lay out those first few building blocks, and then it can come and continue the process of replication. So that's why we start off with primase. Now enters the main character, DNA polymerase. As you can see, it's synthesizing the new strand. So after those first few building blocks, it starts laying out the rest of the nucleotides. Now, let's talk about the directionality of DNA polymerase. So I really wanted to talk about directionality so that you never get a question about the leading versus lagging strands incorrect. So DNA polymerase reads strands from the 3' to 5' direction, and so it synthesizes the new strand from the 5' to 3' direction. And just to really drill in this point, this is the structure of a nucleotide, or at least its backbone. I didn't represent the nitrogenous base because it's not relevant here, but you can see that this is the ribose sugar, and you'll see that at the 5' end you have a phosphate, and at the 3' end you have an OH. You can only add new nucleotides to that OH, the 3' OH. And that's why DNA polymerase will only synthesize new strands from the 5' end to the 3' end. So let's apply this to the replication fork. This strand is running 3' to 5' at this end, and this one's running 5' to 3'. Now, of course, DNA polymerase can only read strands from the 3' to 5' direction. So for this strand, that's not a problem because it's going in the same direction as helicase. So it's going from 3' to 5' here. Whereas with this one, DNA polymerase cannot join right here at its 3' end and start reading like that. And that's why, as we'll see, this will be the lagging strand. So this is the leading strand because at that open end, it starts at 3'. Whereas if we turn our attention to the lagging strand over here, as you can see, DNA polymerase might start somewhere like right here, read this from 3', so this would be its 3' end to 5'. And then as soon as it runs out of nucleotides to replicate, it'll jump back and then read more of the strand, and then jump back again. And then when more opens up, it'll jump again. And that's why you get the Okazaki fragments. So an easy way to identify the leading versus lagging strand, look at the end that's open, so this side, and if it's 3', then you're gonna have the leading strand over there. If it's 5', then you have the lagging strand. Okay, the next enzyme I wanted to talk about is ligase. Ligase will help seal the gaps that result from those Okazaki fragments. It's also used in recombinant DNA and biotechnology. I discussed that at length in my lab techniques playlist, specifically the 4th video, but you'll find it in the description box down below. So those are all the high yield enzymes. One thing I wanted to mention, not very high yield on the MCAT, but these initial primers are actually RNA nucleotides rather than the nucleotides used in DNA. So sometimes you'll see that an enzyme known as RNase will come and remove these nucleotides, and then ligase and DNA polymerase can fill in the gaps over there. But that's not very high yield. With that, we conclude our video on the DNA replication fork. If you found this video helpful, be sure to like and subscribe and share this video with your friends. It really helps the channel grow, and I would really appreciate it. All right, love you all, and I'll see you in the next one.